We should work in like
Hi, everyone.
Sorry.
Okay, now, right. Sorry about that. You all got to see us setting up a little bit. Thank you for your patience. Also, my apologies if you can hear that lovely background noise. But so I'm Lynn Lonis. I'm one of the organizers, and I'm going to be running this afternoon session. As a friendly reminder, if you have a question, you can use the Ask the Question tool to do that, and people can upvote. But without much further ado, I will introduce our first speaker, Signy Sheldon, who will be speaking today about multiple ways to retrieve episodic autobiographical memories, the how and why.
Okay. So let me just, okay. So you can see the screen or the?
Yes, or at least I can see. Great.
Okay, excellent. Yeah, so I'm only able to see the slides right now, but thank you for coming to this virtual talk. It's a real pleasure to be part of the symposium. So far, the talks have been wonderful. So let's keep it up. So as Lynn said, I'm going to be talking to you about multiple ways to retrieve episodic autobiographical memories, how that happens, and also why that occurs. So autobiographical memory retrieval, it requires episodic memory processes to access and associate a whole host of information and details related to a personal past event. For example, if I were to recall a lunch I went to with friends, I'm going to recall general details about that memory, such as things I know about, let's say, the tuna fish sandwich I ate at lunch. And I'm also going to recall more specific imageable details from that event, like where I was, what I saw, and perhaps even the smell of that sandwich. Now, these details are stored separately, so when we recall these memories, we have to bundle them together into a single memory representation. And in the brain, autobiographical memory retrieval depends on a diffuse network of regions that centers on the hippocampus with different details represented by different brain systems. So it's the hippocampus that binds together these access details of a remembered event that are stored across those cortical systems into that memory representation. And as Dr. Schachter illustrated really nicely, this is done flexibly. And for retrieving past memories, this means that different sets of details or systems can be included in that representation at retrieval. So in other words, while the hippocampus is needed for retrieval, how these broader systems are active at the time is going to determine the content of the memory. So the question is, well, what are these systems? So there's neuroimaging evidence as well as some autobiographical theories that we can pull from to try to understand how autobiographical content is processed across the brain. So first, there's this reliable separation of larger scale cortical networks that support retrieving conceptual versus perceptual episodic content from lab-based tests. So this is described really nicely by the PMAT framework developed by Maureen Ritchie and Charan Ranganath, which proposes a posterior medial brain network for processing contextual details and an anterior temporal system for processing more conceptual aspects of episodic memories. Now, this content distinction is also seen in theories of autobiographical knowledge that propose that this knowledge is stored in some sort of specificity hierarchy from general concepts that relate to the gist of a recalled event to the perceptual symbols or images that were present in one specific episode we're recalling. So from this work, we've proposed a neural distinction in how we retrieve or recall conceptual versus contextual content of autobiographical memories. So while conceptual details of when these conceptual details of an autobiographical memory are recalled, so this is recalling details such as, you know, how you felt about that sandwich you ate at that lunch and maybe it changed your views on tuna, that is going to recruit an anterior memory system that includes regions like the temporal pole, the perirhinal cortex, and the ventromedial prefrontal cortex to process these higher order, more evaluative details. So this is what we call the conceptual remembering system. In contrast, when contextual or imageable details are recalled from an event, like remembering sitting next to Fife at a table that had grainy wood from that lunch, a system that engages perceptual processing is going to be active. And this is going to include posterior regions like the precuneus, sensory integration regions, and the parahippocampal gyrus. Now, a bias towards one of these systems is going to be reflected in the content that's used to construct event representations by the hippocampus. So the proposal is that the hippocampus could be the site of integration between these two systems. Now, within the hippocampus, there are indications of functional specializations that do pertain to memory. So there's structural and functional evidence that the posterior hippocampus preferentially supports processing contextual details needed for forming finer grained memory representations. And the anterior hippocampus is important for integrating more diverse types of details, processing the global aspects of an event. Now, I just want to note that this role of the anterior hippocampus has led to some idea that perhaps it plays a more necessary role in initiating constructing autobiographical memory representations as it lays the conceptual groundwork for that construction. Okay, so today I'm going to try to answer two questions about these neural systems and how they perhaps relate to the hippocampus for autobiographical memory-like tasks. The first is to see if there are separate brain systems involved in accessing conceptual versus contextual details of autobiographical memories. And the second explores the functions of these systems, testing how they apply to cognitive scenarios outside of remembering. And here I'm going to look at personal problem solving. Okay, so I'll start with this first question. So to answer this question, my graduate student, Lori Gagurian, and I ran an fMRI study in which we biased people to recall conceptual or contextual details of their own memories. So before scanning, we had each of our 24 participants give us a list of personal memories and make event captions for them, something like meeting Leonard Cohen, and we used those as stimuli in the scanner. Now, all these memories were rated and equated for subjective measures of remembering, and we asked for both recent and remote memories. So this is based on some questions about a potential different neural support for retrieving memories from these different time points. So that's before the scanner. In the scanner, we showed the participants their event captions a few times under different experimental trials. So all trials began with retrieving a memory with all the details they could recall, and then they entered into this reorienting phase that differed for the conceptual versus contextual trials. For the conceptual trials, participants were asked to focus only on the thematic elements of this memory. For a memory like meeting Leonard Cohen, this would mean you would think about why it was important to meet him, what lessons he taught you, and so forth. And then for the contextual trials, participants oriented to the spatial perceptual elements of that memory, perhaps thinking about exactly where they were when they met Leonard and the details of the room. So each trial then ended with participants using those conceptual or contextual details as a cue to access a new related memory. And this is important because it allowed us to look at how activating these systems during retrieval could bias subsequent memory access. So this actually follows nicely on some of the ideas that have been brought up in previous talks. So we used partial least squares to compare and contrast the neural activity as a function of these experimental manipulations. So essentially, PLS gives data-driven significant patterns of activation that dissociate between your manipulations called latent variables. And this analysis resulted in two significant latent variables. So up here on the slide, you see a latent variable that showed neural activity that distinguished between the conceptual and the contextual trials for both recent and remote memories during that reorienting phase. And these distinctions, which are illustrated in those brain figures, really do align with our predictions. So in the blue, that's the brain pattern associated with the conceptual trials. You see regions like the ventral medial and ventrolateral prefrontal cortex, a lot of temporal activity, insula, and also you do see some midline posterior region involvement here. Now, the pattern associated with the contextual remembering trials shown in red included more posterior regions like the precuneus, the parahippocampal gyrus, and the occipital cortex. So again, this does map onto our proposed conceptual and contextual remembering systems. Now, with regards to the hippocampus, we see some preferential engagement of the posterior hippocampus from a parahippocampal cluster during the contextual trials, but there was no difference in anterior hippocampal activity between these conceptual or contextual trials. Now, when we looked at how those conceptual and contextual neural patterns were reflected when accessing that new related memory by these detail types, we see something very interesting. So what we did is we plotted the neural activity associated with these patterns over time, starting when the reorienting phase began to the end of elaborating on that newly cued memory. And what you should notice is, once again, there's no difference between recent and remote memories within each condition. And that the reorienting difference, or that neural pattern, remained when people had to access or think about a new memory. So this suggests that focusing on conceptual versus contextual details is going to affect neural engagement when trying to think of a new related memory. Also from this plot, you should notice that the only pattern for the contextual trials is maintained when thinking about the details of those related memories, suggesting that those context details are more similarly processed in the brain across related events. Okay, so just quickly, over and above that relation between the reorienting and the access phase that I just discussed, we also do find patterns that distinguish between them regardless of the conceptual and contextual trials. So this is the second latent variable that resulted from PLS. And what I want to call your attention to is the pattern associated with retrieving those related memories, which is shown in red. And this included areas in some midline frontal areas, but interestingly, this is where we see actually involvement of the anterior hippocampus, suggesting that perhaps the anterior hippocampus is really important in initiating general memory construction. So in answering this first question, it seems that there are separate brain systems for conceptual and contextual remembering, and the engagement of these systems can vary with retrieval demands. These distinctions, they partly map onto hippocampal functional specialization. We saw that the posterior hippocampus is involved in contextual rather than, or more involved in contextual than conceptual detail retrieval. And the anterior hippocampus is involved, or seems to be involved, in initiating memory formation, regardless of how that memory is formed.
And you have 5 minutes left.
Okay, so the second question I just want to touch on is how these apply to cognitive scenarios outside the domain of remembering. So Dan Schachter illustrated very nicely in his talk that there's clear overlap between the core network that supports autobiographical retrieval and several other tasks that require mentally simulating contextual events. So again, the idea is we can flexibly recombine autobiographical information or content to construct imagined scenarios. So we want to see how this applies maybe to our conceptual and contextual systems. And we have some behavioral support that you actually can shift access to conceptual and contextual details differently for imagined events, just like for remembering. So these are some results that I won't go too far into, in which we ask participants to describe episodic simulations in response to a conceptual event cue or a contextual or spatial cue. And when we looked at the type of episodic details they used to form those simulations, we found that contextual cues biased participants to access perceptual details, whereas those general event cues biased people towards more of what we call story or gist-based conceptual details. But I just want to take the last few moments to focus on how we think autobiographical, these remembering systems relate to one particular future-oriented task, which is open-ended problem solving. So for these types of problems, like these are personal problems like trying to figure out how to host a dinner or how to prepare for an online talk, there isn't semanticized or a scripted solution associated with them. So we have to engage episodic memory processes to mentally construct or simulate possible outcomes. And we found evidence that that episodic memory is important for doing this. So we've tested groups with impaired episodic memory function, including older adults, and we find that they are impaired at describing effective solutions to these problems. So they have fewer relevant means or steps, and they have fewer episodically specific internal details. Now, for autobiographical memory and personal problem solving, what we hypothesize is that conceptual and contextual systems are going to be engaged at different phases that are common to these tasks. So remembering and problem solving begin when a cue activates the generation of potential relevant memories you could retrieve or solutions. And then once one event or solution is settled upon from these potential outcomes, a person is going to stimulate it in detail. So this is what I'm going to call a generative and elaborative phase of retrieval. And we think the earlier generative phase requires access to more conceptual autobiographical information because you have to form these general event representations and compare them, whereas the later elaborative phase requires accessing perceptual processes to processing to bring context to an event simulation. So we ran an fMRI study in which we scanned participants as they did a memory and personal problem-solving task that include these two phases. So all participants all trials began with the general event or problem cue to prompt access to multiply conceptually linked events. A memory cue could be times you're celebrating or exercising, and a general problem cue could be you burnt food at a dinner party. So you've got to think about what you would do. So we asked people to think about multiple memories or solutions to these cues, and then pick one and elaborate on that memory in detail. So we have this generation and elaboration phase. And again, we turn to PLS to compare and contrast neural activity during these phases of memory and problem solving. And we got two significant latent variables. This first one you see up on the slide actually shows separate patterns for memory and problem solving during that generation phase. And we didn't see any distinctions during the elaboration phase. So in red, you see the memory trials preferentially recruits regions like the ventral medial prefrontal cortex and the PCC. Whereas in blue, you see the problem-solving task when you're thinking of many potential solutions. Now you see is more robustly recruiting the ventrolateral prefrontal cortex and bilateral temporal regions. Now, what's more interesting is that the second latent variable shows commonalities between memory and problem solving within each phase, and these align with our predictions. So here we see during the elaboration phase, shown in red, preferential activity in the dorsolateral prefrontal regions, but mainly there's robust activity in the PCC and anterior parietal regions. The generation phase shown there in blue was associated with activity in, there's some activity in ventral regions of the posterior cingulate cortex, but mostly you see preferential recruitment of ventral frontal regions in the bilateral lateral temporal cortex. And it's only during this generation phase that we actually see common recruitment of the hippocampus, and specifically it's in the anterior hippocampus. So that fits with some of our findings from before. So for the second question, what we see is that both for memory and problem solving, there seems to be common engagement of a contextual system to guide mental simulation and a conceptual system to guide accessing and relating event representations to start a task. And bringing these the answer to these questions together, the take-home message is that while there are multiple ways to represent autobiographical pasts and our hypothetical events that emerge from the from activation of the regions within these different memory systems. And we can think about how these memory systems might help serve different purposes and then affect the way that we mentally experience these representations. So perhaps the conceptual representation is going to help us with evaluating and generalizing episodes, and these perceptual simulations, they're going to be useful for formulating very specific plans or when you need to discriminate between episodes, let's say. So from there, I think there's a lot of interesting questions that, or questions that we can consider. For one, we're interested in how these neural systems might underlie individual differences in autobiographical memory retrieval, and whether they're also apparent and these differences are apparent in approaches to problem solving. They also, I think, really do reinforce this idea that autobiographical representations can be formed anywhere along this continuum of content. So, you know, from facts to percepts. And it really is about where you land on that continuum that's going to determine your current thinking. All right, so with that, thank you for listening. And I want to thank the students on this project and the funding agencies.
Great, thank you so much. So I think from that we will jump right into our discussant, Matthew Grilli at the University of Arizona.
Sorry, I gotta. Am I sharing?
Your screen is still being shared, and now it's
okay. Cool.
Thanks everyone for your patience. We'll just take a moment to allow this to load. Hi.
Hello. So I do have some slides. Is it okay
Great.
if I?
Yes. Please share those.
Okay, can you see those slides okay?
Yes.
All right. That's all I can see. So I'm sorry.
Yeah.
Something happens where I get off track. Thank you to the committee for the opportunity to serve as the discussant for Dr. Sheldon's presentation. I, like Dr. Sheldon, study autobiographical memory using cognitive neuroscience and neuropsychological approaches, so it's exciting to be able to build off her recent findings. In this discussion setup, I'm going to focus on this conceptual versus contextual distinction that Dr. Sheldon introduces, and I want to do so at both a cognitive and a neural level, both of which are depicted here from a few recent papers out of Dr. Sheldon's lab. In the first part, I want to make a few connections between this framework and recent research that's been broadly focused on this idea of episodic and semantic memory overlap, the possibility that we can get a glimpse into the higher order organization of autobiographical memory, and questions about what's an item versus a scene when we turn to sort of complex stimuli like autobiographical memory. In the second part, I'll briefly present some neuropsychological data that I think nicely speak to Dr. Sheldon's framework, and then I'll close by raising a few questions, although I know this group can come up with their own. So one area I think that Dr. Sheldon's work has important implications, and she mentioned this towards the end of her talk, is for this ongoing debate about the separation or lack thereof between episodic memory and semantic memory. In particular, Dr. Sheldon's data has certainly raised the possibility that anterior temporal lobe regions have a larger role in episodic remembering than we might have appreciated. Now, it has long been recognized that anterior temporal lobe regions likely have a role in autobiographical memory, but it hasn't been clear whether this is because we need semantic memory to sort of set the background or the scene for episodic remembering, or if anterior temporal lobe regions might provide direct access to episodic details and further elaboration of them. Dr. Sheldon's findings certainly raised the possibility that anterior temporal lobe regions are involved and support episodic remembering. I think this will be an important question for further follow-up. Now, this sort of conclusion would be relevant to recent theoretical work, including work by Dr. Louis Renaud, shown here, and colleagues, which has shown considerable overlap between the so-called core episodic recollection network and the semantic memory network that's been shown through a variety of means. I'll also suggest that Dr. Sheldon's data on conceptual remembering may provide a glimpse into the higher order organization of autobiographical memory. What I mean by this is, how does the brain organize a lifetime of autobiographical memories and allow us to see the connections between them? An earlier study done by Dr. Arnaud Darchambault gets at this question by scanning individuals while they retrieve self-defining memories in one of two ways. They were either asked to focus on engaging in what they called autobiographical reasoning, which involved reflecting on the broader importance of that memory to their sense of self and how it sort of fit into the life story that was unfolding at that period of time in life. And on other trials, they retrieved the self-defining memories by focusing on the specific episodic details, or more traditional episodic remembering type of orienting task. Now, similar to Dr. Sheldon and colleagues, they found an anterior to posterior shift, in their case for reasoning versus remembering. Darshan has gone on in recent work to suggest that this kind of gives us an idea of what it's like to zoom out on one autobiographical event. So I think it'll be interesting in future work to see the degree to which conceptual remembering, while focused on a singular event, could be reflecting or peeking into this higher order organization. Finally, I think Dr. Sheldon's data provides some interesting insights into the organization of what you can call the contents that will ultimately populate an autobiographical memory. And she mentions this as well. Now, it's not presented as an item versus scene comparison per se, but the conceptual remembering versus contextual remembering framework has some similarities. So with conceptual remembering, one could argue that this sort of type of processing might preference people towards objects, people and emotions, the types of things that an anterior temporal lobe network has been hypothesized to support. Whereas contextual remembering seems to be similar to this idea that posterior medial lobe regions might be essential for forming situation models built up out of spatial temporal details about a scene. So this conceptual versus contextual framework may have some important hints into functional specialization as well. So I'll suggest that Dr. Sheldon's framework also has some intriguing implications for neuropsychology. Take, for instance, the question of whether patients who have isolated or disproportionate damage to either anterior temporal or posterior medial lobe regions, will they show a particular type of difficulty in how they try to remember life events? Well, if we consider studies that have documented and carefully scored autobiographical memories using approaches like Brian Levine and colleagues' autobiographical interview, I think we can say that there's some preliminary evidence that things might in fact dissociate to a degree along a contextual versus conceptual framework. Take, for instance, this recent study by Ahmed and colleagues, which I think leads to a posterior medial to contextual remembering idea. Here they compared two neuropsychological groups, individuals with posterior cortical atrophy and individuals with typical Alzheimer's disease, to controls, and they had them retrieve autobiographical memories, and then they scored these memories for the types of details that they generated. Now, what's critical here is the posterior cortical atrophy group had disproportionate dementia-related atrophy in posterior medial lobe regions relative to both the Alzheimer's group and, of course, to the controls. And what they found was that in this posterior cortical atrophy group, there was a selective disruption or reduction in the ability to retrieve spatial, temporal, and perceptual details, the very types of details that Dr. Sheldon suggests might be part of contextual remembering. They further showed that dementia-related atrophy in right precuneus was associated with how much difficulty these individuals had with retrieving one of these types of details, perceptual details specifically. So this is one of several studies that show similar findings. Now, okay, so there's some evidence for contextual remembering in a posterior medial regions of the autobiographical memory network. What about the double dissociation? Do we see evidence for contextual remembering in anterior temporal lobe regions? Here, it's probably fair to say that there's less direct evidence, although there is some evidence that anterior temporal lobe regions seem to have a stronger role in conceptual autobiographical details and content relative to more spatial temporal and contents in episodic memories. Semantic dementia and what we find in that population is a good example of this. You can also see this in a recent study from my lab where we investigated autobiographical memory in an individual MK who developed autobiographical memory impairment after a left anterior temporal lobe resection secondary to epilepsy. The surgical resection took about the 4 most centimeters, anterior centimeters. Interestingly, MK exhibited a disproportionate impairment in the ability to retrieve conceptual autobiographical knowledge, personal facts when he was trying to elaborate on autobiographical events, relative to his ability to come up with spatial temporal details when trying to elaborate on those memories. And that contrast is shown here. In both cases, he's impaired, but when directly compared, he was more impaired for conceptual autobiographical details.
And if you want to try to wrap up in the next minute or so.
Can do. So we've made some connections to current topics. Where might we go from here? I think one thing to consider is the nature of conceptual and contextual remembering. For instance, as I've alluded to and Dr. Sheldon has too, thinking broadly about themes of an event and emotions while conceptual remembering may naturally draw attention to people, objects, and the emotions that we have in relation to them. If this is true, we could ask whether we might see stronger evidence of conceptual remembering in posterior medial regions if we emphasize themes about place and time. Likewise, we might see more evidence of contextual remembering in anterior temporal regions if the focus for perceptual details was about objects and people. We also could consider what factors might alter the balance between conceptual and contextual remembering. So some memories may naturally be more conceptual, and there is some evidence that more remote memories do become semanticized like this. You could also ask whether the commonly reported anterior shift in remembering for older adults might reflect a move towards conceptual remembering. I think this is an interesting question for future work. And part of this will probably be developing narrative scoring protocols like the autobiographical interview, but are more geared towards evaluating conceptual versus contextual details. And these future directions aside, Dr. Sheldon's framework clearly makes important connections to a variety of topics that are ongoing in cognitive neuroscience, and it's a valuable framework for thinking about the complexities and applications of autobiographical memory. Thank you.
Great, thank you so much. Okay, so for the sake of time, I think we're gonna have a shortened question and answer period. But the question that, so we have 3 questions. The one that got the most votes was by Miriam Ali that says, you showed an anterior conceptual versus posterior contextual distinction very nicely. I'm trying to think through how that relates to another distinction you mentioned between anterior systems being more gist-based and posterior systems being more detail-oriented. In most of your tasks, you are looking at fairly detailed memories and problem-solving scenarios. Would your observed results change if you had people orient to very general or very generic concepts or concepts?
I think that's a really interesting question. If we had people orient towards the general concepts, we tried to do that in the scanner with the memories, but whether we would see that, let's say, for problem solving, I think is a very good question. So how this relates to the distinction between the gist versus the detailed orientation, I think that's a really important question. And I think Matt alluded to that, whether these distinctions in these anterior versus posterior systems are reflecting content or specificity. So I think in order to get at that, what we would have to do is perhaps have a study in which we got people to think about conceptual versus contextual information and then vary the level of specificity with which we would get them to think of. And my feeling is that it really is about the content rather than the specificity of what is being retrieved. But inherently, a lot of these contextual details seem specific in how we define them.
Thanks. Yeah, I think it's a really interesting thing to consider further. And so for the sake of time, I'm going to ask you one other question. I'm also sort of getting at how you're defining these dimensions. The votes keep changing, but this is from Boudica Belana. And so he says, great talk. I'm curious about the conceptual versus contextual continuum. Having conceptual knowledge can act as a scaffold to support remembering perceptual details. So how does that fit into these systems being at 2 opposite ends of a spectrum or continuum?
Yeah, that's a great question. And I think, this is something that, those continuum models have to take into account that perhaps it's not like this or that, or it's not conceptual or contextual. And I think this is why we see there are some issues in trying to find that double dissociation. So one idea is that conceptual knowledge might be something in which the groundwork for then forming a representation it is the groundwork for that. And then you add on details within the contextual remembering system. And that could be why we didn't see any differences in the anterior hippocampus or any greater involvement of the anterior hippocampus for the conceptual remembering. So I think it would be interesting, though, to apply this information to different ways in which people are cued for memory. So you can think that there are some memories that are very much cued by very specific context and details, and they just really flash in one's mental representation. So it would be interesting to devise a way to look at the cueing and if we would see some differences.
Interesting, thank you. Okay, so I think for the sake of time, we're gonna move on now to our next talk, but again, as I'm sure you can see in the chat box now, there's a lot of round of applause, and I'll give you another round of applause as well to both of you. Thank you very much. And so next up is Molly Hermiller talking about hippocampal-targeted theta-patterned stimulation, which immediately enhances hippocampal memory processing, which is a simultaneous TMS fMRI experiment.
Hi.
Hi. Okay, great. So we can all see your slides. I'm not sure what it looks like at your end, but.
Okay, you can hear me okay?
Yes.
All right, I'll go ahead and get started. Hello, everybody. Thank you for having me. I'm Molly, and today I'll be presenting some results from my main project in grad school where I worked with Joel Voss at Northwestern. And in his lab, we used TMS to study the hippocampus and its network and their role in memory. So I don't think I need to convince anybody here that the hippocampus is important for memory, and it's thought to interact with a set of distributed brain regions, a network that I'll be referring today to as the Hippocampal-Cortical Network, or HCN. And one way we can observe or measure this network is using task-evoked fMRI activity. So this is an example from Neurosynth, the results from using the search term episodic memory. So these are regions that have a strong, bold activity or really light up during episodic memory. So whether that be the encoding or retrieval processes. And these are regions that we typically consider to be part of this network, the HCN. Another way we can observe or measure this network is using resting state connectivity. So again, from Neurosynth, an example here, this is the result of placing a seed in the left hippocampus, and these are regions that show robust connectivity. Again, you can see there's this overlap in these regions, and so this is what we refer to when I'm saying the HCN. And these two fMRI techniques, task evoked and resting state connectivity, these are techniques that I used in the projects I'll be speaking about today that we use to observe and measure the network. And we also use TMS in order to then modulate or experimentally manipulate the network. And so the hippocampus, which we're very interested in, is not a region of the brain that is directly accessible with TMS. So we can't directly stimulate it. TMS really is only stimulating a small set of, not small in terms of, it's about a cubic centimeter of neurons. So depending on what you consider small, but you're not flooding the whole brain. You're not able to stimulate the hippocampus with TMS. So instead we use this HCN, or network targeted stimulation approach, in order to indirectly modulate or influence the hippocampus and its network. And so what we do is, before subjects take part in our studies, we have them come in and we collect a baseline resting state scan. So for example, here's a subject and we then place a seed in their left hippocampus, and we can create this HCN, or this hippocampal connectivity map, for each subject. And then we select a site in their left parietal cortex, and that becomes their stimulation target. And we do this for all the subjects. We have these individualized stimulation targets demonstrated here, where each green sphere is a different subject stimulation site. And so we first started using this HCN targeted approach several years ago in a study where we delivered 5 days of active stimulation, a 20 hertz high frequency stimulation train, a week of that. And then subjects also received 5 days of sham stimulation. And we found that a week of active stimulation significantly improved associative recollection relative to a week of sham stimulation. And this active stimulation also increased connectivity, or the number of links or connections, especially in regions that already showed high baseline hippocampal connectivity to begin with. And we recently replicated the behavioral findings And a group at the NIH has replicated the fMRI findings. And so notably, these fMRI changes, the robust changes occurred between the hippocampus downstream of the site that we stimulated. It was hippocampal connectivity between these posterior medial regions of the network. And notably, these posterior medial regions, in some intracranial studies, they have been shown to synchronize or have theta phase locking values with MTL or hippocampal regions during successful autobiographical or self-episodic memory relative to math or when you're just at rest. And so we think that this is a functionally relevant oscillatory activity pattern in the brain. And so in the study I mentioned previously, we use a 20 hertz or just a high frequency train of stimulation, and we were able to modulate and interact with the network. But if we can somehow interact with the theta pattern in the network, perhaps we would be able to enhance or modulate or interact with the HCN more robustly using TMS. And so there is a TMS protocol, it's called theta-burst, that mimics the endogenous theta rhythm in the hippocampus, the theta nested gamma rhythm, where 50 hertz triplets are delivered every 200 milliseconds or every 5 hertz. And so a few years ago, we ran a project where we compared the effects of a continuous theta burst, intermittent or interrupted theta burst, and then the beta frequency or the 20 hertz protocol that we previously used, as well as a sham. And we compared the effects of these 4 stimulation protocols and within subject design, and we found that the continuous theta burst protocol was the only condition that significantly improved D prime on a word recognition task relative to sham. And this memory enhancement was related to an enhancement or an increase in hippocampal connectivity in this posterior medial region of the network. So to kind of piece everything that we just talked about together, we found that our HCN targeted stimulation approach is one that effectively modulates our network activity. And we found this in a variety of studies from the lab. And the one that I previously mentioned indicated that the theta burst pattern stimulation may be more effective at modulating our hippocampal network. But these studies were all looking at long-term or after effects of stimulation. So this chunk of studies was looking at the 5 consecutive or cumulative effects of 5 consecutive days of TMS, and we're looking at the effects 24 hours after stimulation. Or this study here is looking at the effects of 1 single TMS session, 1 hour post stimulation. And so these studies did not tell us the immediate effects of stimulation. And so in order to do this, we ran the project I'll be talking about for the rest of my time here, which had the overall aim to determine the immediate effect of network-targeted theta burst stimulation on hippocampal or MTL memory processing. So in order to look at the immediate effect of TMS, we delivered stimulation inside the scanner, and these are just some example photos of what the setup looks like here. And then we developed some scan sequences, one that allowed us to deliver theta burst stimulation during scanning, and one that allowed us to deliver a 12.5 or an alpha or beta, however you want to define that, frequency stimulation during scanning. And so the scan parameters were designed and set up so that the TMS pulses indicated in the colored lines here would not interfere with our scan acquisition. And then the 12.5 hertz or alpha frequency serves as a control frequency to our theta burst condition of interest. And so while they're in the scanner being stimulated and scanned, they are performing this task. And so for some of the trials, they are seeing a scene, an outdoor complex visual scene. And for other trials, they are just seeing a number. And so while the scene is on the screen for the 2 seconds it's there, subjects are instructed to really immerse themselves in the scene. Imagine what kind of activities they could do there, what the weather would feel like, whether they've been to a place like that before or not. Just really immerse themselves in that scene. And this kind of encoding, it's an intentional encoding task. So they knew when they came out of the scanner, they would be asked to, they would be tested on their ability to remember that scene or not. So this intentional encoding and scene processing is known to evoke a strong bold signal in the hippocampus. Whereas if it was a trial that had a number on the screen, then the subjects were just instructed to indicate if it was an odd or even number. And so this kind of numerical judgment is known to not evoke a bold activity in the hippocampus. And then for some of the scenes in the 2 seconds preceding the stimulus onset, TMS was delivered. And then for another subset of trials, in the 2 seconds preceding the stimulus onset, stimulation was not delivered. And so therefore we had an off or a non-stimulation control. And again, when the subjects knew that when they came out of the scanner, they would be tested on their ability to remember or distinguish the scenes from blurs using this remember, familiar, new paradigm. And we were particularly interested in scenes that were later recollected, so endorsed with a remember response. So when we could then backsort all of our fMRI trials based on their subsequent memory performance, and we suspected that scenes that were later endorsed with a recollection or remember would have the greater bold activity relative to familiar or new scenes.
And you have about a minute left.
Okay, thanks. And so just to sum this up, subjects came in for 2 different sessions where they received either TBS or alpha, and stimulation was delivered to an HCN site in the parietal cortex or out of network location in the supplementary motor area. And these conditions were counterbalanced. They saw scenes and numbers with TMS on versus off, and they were tested after the session. And we predicted that HCN TBS would uniquely influence hippocampal MTL processing. And the idea here is that HCN TBS would cause successful entrainment in the hippocampus, so that there was an in phase or an entrainment phase locking within the hippocampus, so that when the scene turned on and the bold response would be activated, would be all in phase. Whereas scenes that were not entrained due to the control conditions would not have that same increased power. Whereas numbers would not really care if they were entrained or not, because there wouldn't be a strong hippocampal activation. And we had 5 ROIs in the longitudinal axis of the hippocampus where we extracted our fMRI activity during scenes that were later recollected. And we found that the 2 most anterior ROIs here had a significant effect of condition. And this was driven by the HCN theta-burst having more robust or a greater signal. There was no effect when we looked at the same ROIs in the numbers. And in the right hippocampus, which is non-targeted, all the TMS was delivered in the left hemisphere. There was no significant effective condition. And real quick with the behavior, we are again interested in scenes that were later recollected. And we found when we compared the 2 sessions, the HCN and the SMA conditions, there was a significant effect of the stimulation type. So whether it was TBS or alpha, and if it was on or off. And when we just look at all of the pairwise conditions, the HCN TBS was the only condition that showed either a trend or a significant pairwise difference relative to all of the other control conditions. And so in conclusion, the HCN targeted theta burst increased evoked activity during hippocampal MTL processing and enhanced memory formation. And this provides causal evidence for the functional role of theta in the hippocampus and its network. And I just want to quickly thank my lab members, people at our imaging center, and all the funding before I take questions.
Great, thank you so much. So for the sake of time, I think I'm just going to ask you one question that was upvoted the most. So this question is by John Saccone, and it's more about the methods, asking, isn't the reason people typically have not looked at immediate TMS effects is because of the increased excitability from stimulation, in particular theta burst, in the next tens of minutes? And was there any way to account for this?
So we were looking we were just delivering a 2-second volley rather than a full TMS train and looking at the after effects. We were delivering just a 2-second volley and then looking at about 8 to 14 seconds right after that TMS train. So we were looking at the long lasting effects of it. And then we also did look for cumulative effects. So to see if there was an effect immediately on the trial immediately afterwards. We did not see any cumulative effects. But of course, that's still up for, there could be a whole experiment designed around answering that question alone.
Great, thank you. So I think we will leave it in that and then for the sake of time, move on to the next talk. But thank you again, that was really interesting. Right, so up next, we have Lukas Kunz, who's going to be talking about anchor cells in human medial temporal lobe and how they represent egocentric directions using during spatial navigation. Sorry. Hi.
Hi.
Right, so if you just want to share your slides.
Can you see my slides okay?
Let's just give it 1 second. Yes, great.
Okay. So, hi everybody. My name is Lukas Kunz. I'm a postdoc at the Freiburg Epilepsy Center in Germany. I'm very pleased to give the next talk on anchor cells in the human medial temporal lobe, and I would like to thank the organizers for giving me the opportunity to present here. In my talk, I will address two main questions. First, what are actually anchor cells, and what are the characteristics of anchor cells? And second, I will address the question, what's the role of anchor cells in episodic memory recall? There are two main reference systems to accomplish successful spatial navigation. On the one hand, there's the allocentric reference system shown on the left-hand side. And on the other hand, there's the egocentric reference system. In the allocentric reference system, spatial information is encoded in relation to the external environment. For example, this blue spot here can be said to be located in the northwest part of the environment. In contrast, in egocentric reference systems, spatial information is encoded in relation to the navigating subject. In this case, the blue spot here on the right-hand side is said to be located ahead and several meters away from the navigating subject. Over the past 50 years, there has been much research on the neural basis of the allocentric reference system, which has led to important discoveries such as of place cells, head direction cells, and grid cells. For example, place cells shown here in the upper row activate whenever an animal or a human is in a particular location of an environment. In contrast, only recently a few studies in rodents started to elucidate the neural mechanisms of the egocentric reference system. For example, there are so-called goal vector cells shown here in the first row, discovered in the hippocampus of flying bats. And in this example here, the goal vector cell activates whenever a spatial goal is ahead of the flying bat. Second, there have been item-bearing cells identified in the lateral-anterior cortex of navigating rodents, and these cells activate when external items such as objects are in a specific egocentric relation to the navigating rodent. And thirdly, last year, center-bearing cells in the rodent posterior cortex have been identified, which activate as a function of the egocentric direction of the center of the environment relative to the navigating rodent. In this case, for example, shown in the lower row of this slide, the center-bearing cell activates whenever the rodent navigates away from the center of the environment. In humans, basically the single neuron mechanisms of the egocentric reference system are unknown. However, it is important to unravel both the neural mechanisms of allocentric spatial representations and egocentric spatial representations, not only from a pure spatial navigation perspective, but also from the memory perspective, because influential theories propose that egocentric neural representations provide also the neural basis for episodic memories, whereas allocentric spatial representations provide the neural basis for semantic memories. Hence, in this study, we aimed at identifying neurons in the human MTL that encode the spatial information in an egocentric reference frame and to examine their role during episodic memory recall. To do this, we performed single neuron recordings in epilepsy patients. In the first study, we had a group of 14 epilepsy patients recruited at the Freiburg Epilepsy Center in Germany, and we recorded 729 neurons in 18 different experimental sessions. The neurons were recorded from different MTL regions, including amygdala, anterior cortex, hippocampus, parahippocampal cortex, and temporal pole. While we recorded the single neuron activity, the epilepsy patients performed an object location memory task in a virtual environment, which is depicted here. After an initial learning session in which the patients learned the locations of 8 different objects, the patients then performed multiple test trials. In each test trial, they were cued with one of the 8 different objects. They then tried to remember and navigate towards the associated object location. They received feedback depending on response accuracy and then re-encoded by collecting the object from its correct position. We identified anchor cells by analyzing the single neuron activity as a function of the spatial behavior of the participants in the virtual environment. Specifically, we stepped through multiple different candidate anchor points, here shown as gray dots. For each of these candidate anchor points, we estimated the tuning curve of the cell as a function of the egocentric direction of this candidate anchor point relative to the navigating subject. Using surrogate statistics and cluster-based permutation testing, we then identified a contiguous cluster of significant candidate anchor points, shown here in light blue. And this contiguous cluster of significant candidate anchor points then constitutes the so-called anchor field, and the center of mass of this anchor field is the anchor point. Here are two example anchor cells from two different patients. In the upper row, you can see one example anchor cell, which has its anchor point in the southeast part of the environment. The anchor point is shown as the color dot with a black small circle around it. And this anchor cell here activates whenever this part of the environment is right ahead of the navigating subject, which is shown here in the middle part of the upper row. Another example anchor cell is shown in the lower row. You can see that the anchor point of this anchor cell is located in the center of the environment. And here you can see that this cell activated whenever the subject navigated away from the center of the environment. We then characterized the anchor cells in our dataset and found that they were particularly prevalent in human parahippocampal cortex, here with a percentage of about 30%. We found that some anchor cells also exhibited tuning of allocentric direction cells and place-like cells, but a significant number of pure anchor cells remained when we excluded those anchor cells that showed either direction cell activity or place-like cell activity. In panel C, you can see the distribution, the spatial distribution of the anchor points, and you can see that they were located both in the center of the environment and also in the periphery of the environment. In panel E, you can see the circular distribution of the preferred anchor bearings. For center anchor points, we observed that the anchor cells preferred anchor bearings of ahead and behind, whereas for periphery anchor points, there was a roughly uniform distribution of preferred anchor bearings in our anchor cells. Importantly, which is shown in F and G, is that a significant number of our anchor cells also showed distance tuning towards the anchor point. So, for example, in F, you can see that this anchor cell increased its neural firing rates whenever the subject was far away from the anchor point. In contrast, in the example in G, you can see that this anchor cell increased its activity when the subject was close to the anchor point. This is important because it establishes a complete full vectorial representation of space in an egocentric reference frame. After having identified anchor cells in the human MTL, we then aimed at identifying their role in episodic memory recall. To this end, we conducted a separate single neuron recording study, again performed at the Freiburg Epilepsy Center in Germany. In this study, 12 epilepsy patients participate and we recorded 737 neurons in 20 different experimental sessions. Again, we recorded single neuron activity from different regions of the MTL. In this case, the participants performed a hybrid navigation memory task called Treasure Hunt, which was developed by the Jacobs Lab at Columbia University. This task had the advantage that there were separate periods for navigation and memory. And I will guide you through the different periods of this task. In one, you can see that at the beginning of a given trial, and the participants performed multiple trials of this task. So at the beginning, the participants were located at a random position on a beach in a virtual environment. They then navigated to several different treasure chests that were scattered throughout the environment. Upon arrival, the chests opened and revealed a unique object, and it was the task of the participants to remember the unique objects and the associated locations of the treasure chests. In step 3, the participants were then transported passively to an elevated recall position. From there, they had then to perform, shown in 5 and 6, 2 different types of memory recall. The first type was location-cued object recall, and the second type was object-cued location recall. Regarding the former, a specific location on the beach was shown, indicated here by the blue circle, and the patients had to recall the associated object that was found in the chest positioned at that location. In
We have
contrast,
about 1 minute left.
yeah, I will come to conclusion. In the second type of recall, an object was shown to the participants, and the participants had to move a cursor across the beach to find the associated location of the object. We again identified anchor cells in different MTL regions, for example, this one here in the upper row, which was tuned to an anchor point in the west part of the environment, and this cell activated whenever this anchor point was to the left of the navigating subject. We then analyzed an anchor cell activity during the two different types of memory recall. And during new patient-viewed object recall, we found that anchor cells particularly activated, which is shown in green, during a successful memory recall as compared to unsuccessful memory recall. This effect was not seen in non-anchor cells. This means that there's an early activation of anchor cells during successful location-viewed object recall. On the other hand, during object-viewed location recall, we found that anchor cells particularly increased their firing rates shortly before the response of the subjects during successful memory recall as compared to unsuccessful memory recall. This effect was not seen in non-anchor cells. This means that there's a late activation of anchor cells during successful object-viewed location recall. To summarize, in this study, we described anchor cells in the human MTL that encodes the egocentric direction towards local reference points, which are termed anchor points. Anchor cells were most prevalent in perihippocampal cortex, and anchor points were positioned in various parts of the environment with a slight overrepresentation of the center of the environment. Some anchor cells showed additionally distance coding, providing a full vectorial representation of space and egocentric coordinates. During episodic memory recall, we found that there was an early activity increase during location-viewed object recall and late activity increase during object-viewed location recall. This is relevant in several respects. We proposed that anchor cells may support egocentric spatial maps in general, that anchor cells may support transformation of perceptual input into allocentric spatial representations, that they may also support the retrieval of egocentric spatial context during episodic memory recall, and that they finally may support the planning of future scenarios from an egocentric viewpoint. With this, I would like to thank all my collaborators, including Andreas Schulze-Bohnagel, Josh Jacobs, Christoph Weidemann, Maike Hahner, Nora Herweg, and Richard Kempter. And thanks to you for your attention.
Great, thank you so much. So I guess I'll give it just another moment to see if anyone else has any questions that they want to ask during this Q&A period. Oh, okay. A question came from Miriam Ali. You mentioned that many cells are relatively pure anchor cells and that they are not also place cells or head direction cells. Can you also rule out that they are spatial view cells? She said, I'm having trouble distinguishing between how a spatial view cell would respond differently than an anchor cell.
Mm-hmm. So I would assume that you are referring to spatial view cells discovered in monkeys, like in the studies from Rolls et al., for example, Hippocampus 1991. These spatial view cells are mainly allocentric neural representations and not egocentric neural representations. And they are also particularly tuned to the walls of an environment. So they mainly increase their activity when a monkey looks at a specific part of, for example, a wall. Yeah. So there could be some overlap between spatial view cells and anchor cells. I would think that anchor cells are then a more general type of neuron, with spatial view cells being a subtype, because the anchor cells can have their anchor points in various parts of the environment.
Thanks. I'll ask one other quick question. One other quick question. What do you think the relationship is between anchor cell representations and hippocampal and entorhinal representations?
Okay, so I think there are many interrelationships. If you're, for example, referring to place cells, one could assume that the combination of input from anchor cells and allocentric direction cells leads to the existence of place cells, which has been, for example, put forward in a review by Bichansky and Burgess in eLife 2018. With regard to entorhinal representations, for example, these item-bearing cells, which were discovered in 2018 by Wang et al. In Science, there might be some overlap, although it seems that these item-bearing cells are preferentially activated by external items such as objects. In our case, we couldn't find that the anchor points were significantly close to, for example, the objects. So I think that in line with the centroid model from O'Keefe in 1991, anchor cells can have anchor points in various parts of the environment, which are not specifically designated, for example, by the presence of an object.
Thank you. And I feel I should also give credit that was Aya Amaminadad's question. But I think for the sake of time, we will now move on to the next talk. But thank you again for a really interesting and fascinating talk. And right, I'll give you a round of applause, as you can probably also see from the chat. So up next is Nora Herweg, who will be talking about multi-unit activity in human MTL and how that reflects retrieval of spatial and temporal context.
Hey everyone, let me just quickly share my slides. All right. Cool. So I'm Nora Herweg. I'm a postdoc working with Michael Kahana at the University of Pennsylvania, and I'm going to be talking about multi-unit activity in the human MTL related to contextual memory. In the first part of my talk, I'm going to present the idea that we can use recall sequences during free recall to infer or assess contextual memory. And in the second part, I'm going to talk about neural activity in the MTL that is related to successful contextual encoding and retrieval. In a standard free recall task, the subject encodes a series of words shown on a computer screen one at a time, and then subsequently attempts to recall as many words as possible. And in this scenario, we cannot only look at whether or not a certain item was correctly retrieved, but we can also look at the order in which the items are remembered. So in this example, frame was presented right after cup. So if those two items are being recalled successively, we would call that a temporal lag of +1. And house was presented 2 before frame, so that would be a temporal lag of -2. And if we analyze the data in this way, what we typically find is that people tend to recall items together that were presented in temporal proximity. This is called the temporal contiguity effect, and it can be seen in the lag CRP curve as a high probability of transitioning between nearby items. What this suggests is that during recall, we can retrieve with each item the temporal context that was associated with that item during study, which then cues other items that were encoded in the same context. However, out in the real world, our memories are not only associated with a particular temporal context, but they can also be associated with a particular spatial context. And to study this, we use the courier task, which is a free recall variant where the encoding phase takes place in a virtual city. It's called courier task because the cover task for the subject is to be a bike courier and deliver objects to different stores in the city. And I'm going to be showing you a brief video so that you can understand what that looks like. So here you're going to see the subject navigate through the city. On every list, there are 12 items that have to be delivered to 12 different stores. Here the subject searches for the florist, and once they arrive, they hear the word daisies, which is the object that they're supposed to remember. Prior to each delivery, there's a pointing task where the subject has to point in the correct direction of the next store. That way we can assess memory, but we can also give them feedback so that they don't get lost. One thing to keep in mind is that all the items are semantically related to their target store. So here the subject is driving up to the craft shop and delivers the item glue. Once all the items are delivered, there is a final navigation to a 13th store where no item is delivered. It's kind of like a distractor. And then once the subject arrives at that store, they are supposed to recall all the items that they encoded. And then after the recall phase, the whole process starts over for a new list, and there are multiple lists in one session. Okay, so here I'm showing you an overhead view of the environment with an example list. You can see that now every recall transition is associated not only with a temporal lag, but also with a spatial lag that indicates how far the items were presented apart in the virtual environment. And what we find then is that there still is the well-known temporal contiguity effect, which you can see in the lag CRP on the right. But we also now find a spatial contiguity effect, meaning that subjects tend to recall items together that were encoded in spatial proximity in the city. And what that suggests is that spatial context, like temporal context, is reinstated during recall and cues associated items. We cannot only look at this effect at the aggregate level, but we can also calculate a spatial and temporal clustering score for each individual recall transition. And that lets us infer which of the items were recalled together with their spatial context and which of the items were recalled together with their temporal context. So in this example, the subject transitioned from pliers to retainer. Those two items were encoded immediately next to each other in the environment, so that transition would receive a high spatial clustering score, but a low temporal clustering score because the items were presented on opposite ends of the list. And in contrast to that, transitioning from retainer to flower would get a low spatial clustering score because the items were presented on opposite ends of the environment, but a high temporal clustering score because they were presented right after one another. And we can use these spatial and temporal clustering scores for every recall transition to then understand what is going on in the brain when subjects either retrieve spatial context or if they retrieve temporal context. And we have previously used this method to show that theta power in the medial temporal lobe increases during the retrieval of spatial context. Now, in this current study, we want to look at the neural signature of contextual memory. We recorded from patients who were undergoing clinical seizure monitoring for epilepsy treatment. And these subjects did not only have macroelectrodes, but they also had microwires implanted in the medial temporal lobe. We had a subset of 19 patients with these microwires. They completed 45 sessions, and we recorded from 462 wires. And we high-pass filtered all the data and thresholded it to isolate the spikes of 566 multi-units, which we localized to either the hippocampus, the perihippocampal gyrus, or the amygdala. We then calculated the firing rates of these units for the 1 second time period following the encoding of each word, and for the 1 second time period preceding the recall of each word. And then we used a mixed linear model to relate the clustering scores for each event to the firing rates of these units across events. I'm going to be showing you the results here. In the bottom row, you're going to be seeing changes in firing rate during the recall time window that are associated with spatial and temporal clustering. And in the top row, I'm going to be showing you changes in firing rate during the encoding time window that are associated with spatial and temporal clustering. Every one of these graphs shows the linear effect of the clustering scores on the firing rate. So a positive value indicates that firing rates are high during nearby recall transition, and a value below 0 indicates that firing rates are low during nearby recall transitions. And then the little inset just shows the corresponding regression lines for the beta values. And what we find for spatial context retrieval is that there is a general decrease in firing rates in the MTL whenever spatial context is retrieved. This effect is most pronounced in the hippocampus, but the difference between the brain regions is not significant. For temporal context retrieval, the difference between the brain regions is significant. Here, the decrease in firing rates is specific to the hippocampus, and there is a simultaneous increase in firing rates in the perihippocampal gyrus. And now going to the encoding time window, again, we find a general decrease in firing rates whenever temporal context is successfully encoded. And again, this effect is most pronounced in the hippocampus, but here the effect doesn't differ by brain region. And then for temporal encoding, we see a very similar pattern, but there is no statistically significant effect. So overall, what we can tell from this is that the effect seems to be very similar across all these conditions, and that there always is a decrease in multi-unit firing rates, and that is most pronounced in the hippocampus. And then next, we wanted to know whether the effect that a given unit shows for spatial context encoding or retrieval is related to that same unit's effect for temporal context retrieval, plotted here on the y and the x-axis. And we did find an effect only in the perihippocampal gyrus and only during retrieval. It was a positive relationship, meaning that if a unit responds positively to spatial context retrieval, that same unit tends to also respond with a firing rate increase to temporal context retrieval, which suggests that units in the perihippocampal gyrus don't care what type of information is being retrieved. They are always responding in a similar fashion, suggesting that the perihippocampal response is non-specific to the type of retrieved context. So to summarize that, in the first part, I have shown you that we can use spatial and temporal clustering during recall to infer the retrieval of spatial and temporal context. And then we have used that method to show that there is a decrease in population firing rates in the medial temporal lobe whenever spatial or temporal context is successfully encoded or retrieved. And that decrease is most pronounced in the hippocampus, which aligns well with the idea that the hippocampus holds a sparse code where few neurons fire for individual memories. And then we saw a different response in the perihippocampal gyrus, where there was an increase in firing rates for some of the conditions. And we also saw that the response in the perihippocampal gyrus was independent of the type of retrieved context. And one of the future directions that I'm currently working on is to tie this back in with the prior results, that there are increased theta oscillations during spatial context retrieval, asking how theta oscillations and unit activity relate to each other in this task. Yeah, I'd like to thank the organizers, of course, and then also the lab and everyone who was involved in the project, and I'm happy to answer questions.
Great, thank you so much. So I guess I'll just wait a moment to see if there are any questions that pop up in the Ask a Question box, if anyone has a question. I realize we're running a little bit late. Oh, but actually, okay. So Josh Salat has a question. He says, interesting talk and experimental paradigm. And this might be more of a suggestion or idea than a question. But he was asking if the velocity in the experiment was constant and if that was played around with. So he was wondering if maybe with high velocity, if that might influence, if that might make the temporal lag relatively small, but the spatial lag relatively high.
Yeah, so we didn't change the velocity, and I do think that it's generally interesting to do that. In terms of like the second part of the question, it is already the case that the temporal lag can be small and the spatial lag can be high. And that is because subjects, like we tell them for every delivery exactly where they have to go, and that decorrelates the temporal and the spatial lag, if that makes sense.
Makes sense to me, at least. So I think for the sake of time, since we're running a bit behind and that there aren't any other questions, at least for now, I will leave it there. I will just add before people leave. So after this, so posters are scheduled to start at 3:20. I realized we cut into the break a little bit, but posters are scheduled to go until 4:50. Oh, and I'll just say, yeah, thank you again to Nora for a really interesting talk and really integrating different concepts from CEMS that people are interested in. So, right, posters are scheduled to be next. You should have received an email link to where the each poster has a different Zoom link, so you should be able to view those there, and also the PDFs. And then immediately following the posters, there's also going to be a social event. You should have also received an email with a link to that as well. And so then we will also resume again tomorrow at 11 Eastern Standard Time, but that will also begin with posters. We really won't be coming back to Crowdcast until 12:30 tomorrow. So with that, I will thank you all for joining today, and I hope you enjoy the posters.